Hey everybody, so I'm going to jump right into it for this week's discussion post. All my notes are right down here in front of me, so I apologize if I'm not always looking at the camera. Definitely need some notes for this one. So we're going to talk about the terms prophage and provirus. They refer to different stages in the life cycle of viruses, and they're associated with different types of organisms. So prophages or bacteriophages, I'll define those in a minute, are associated with bacteria, and proviruses are more so associated with um, animal cells. So the difference between a prophage and a provirus. A prophage or bacteriophage is a virus that infects strictly a bacteria. Um, the phage will integrate itself into the DNA or integrate its own DNA into the bacterial host genome. This occurs during the lysogenic cycle of the bacteriophage, and the viral DNA that is there is dormant within the bacterial genome and is replicated along with the host cell host DNA once the bacteria divide. So basically, all of the division that the bacteria does, it as it's dividing, it passes on the virus's DNA to each one of its daughter cells. Prophages can remain dormant or latent in the host cell until certain conditions like stress trigger the lytic cycle where the viral DNA is excised and begins the production of new viruses, leading to the lysis or destruction of the bacterial cell. We'll go more about that later. Proviruses, these are the ones that infect the animal cells. So animal viruses that integrate into the host genome as well. However, this typically occurs in retroviruses. So a classic example would be HIV. This is a provirus that may remain latent for a long period, but the animal cell will not lyse because because of this virus. Does not necessarily enter a lytic cycle where it destroys the host cell. Instead, it can be reactivated at any point to produce new viral particles without killing the host cell. So these proviruses do not do immediate or life-threatening harm to the infected cell. Key difference between the host. Just to recap, the key difference between the host prophage between the prophages and the proviruses is its host. So again, prophages integrate with bacteria, and then proviruses integrate with the animal cells. Proviruses tend to remain in their host cells without necessarily causing immediate cell lysis either. So I'm going to jump down to the latency in animal viruses versus lysogeny in phages. So latency in the animal virus, again, is when the virus remains dormant within the host cell for long periods without producing new viruses or causing any symptoms, and this can reactivate at any time later on down the road. Lysogeny in bacteriophages is a similar process where the viral DNA can be dormant but is replicated within the genome and the DNA of the bacterium as it continues to divide and pass on those cells or pass on that genome to its daughter cells. So eventually, whenever the stress or other certain triggers prompt the prophage to become active again, as that virus reproduces, it eventually causes the bacterial cell to lyse and the release of all of the reproductive viruses. Three differences between the lytic and lysogenic cycles in bacteriophages. So we're going to start out with viral replication. In the lytic cycle, so the lysis cycle, the bacteriophage takes over the bacterial cell machinery immediately upon infection and to produce new virus particles, which eventually leads to that destruction of the cell. In the lysogenic cycle, the bacteriophage integrates its DNA into the host genome and may remain dormant for X period of time. The viral DNA is replicated passively, and the bacterium replicates its own DNA without producing any new viral particles. Number two, the fate of the host cell. So in the lytic cycle, at the end of that cycle, no matter what, the cell is lysed and the cell dies. And when you talk about the lysogenic cycle, the animal cell is relatively unaffected and it continues to live even though those new particles were exported from the original cell. So the animal cell or bacterial cell, it survives in the lysogenic cycle. And then latency. In the lytic cycle does not involve a latent period. So viral replication happens almost immediately after the infection of that cell. And then in the lysogenic cycle, a latent period is involved where the virus can remain inactive in the bacterial genome or the animal cell genome for extended periods of time before reactivating or causing some level of sickness that we can pick up on or cause symptoms. The role of lysogeny in development of pathogenicity. So lysogenic conversion occurs when a prophage integrates into the bacterial cell genome and carries genes that enhance the pathogenicity of the bacteria. Genes encode toxins, enzymes, or other virulent structures that make the bacterium more harmful to humans or other hosts. Lysogenic conversion contributing to pathogenicity. I have two examples here. I have the organism that causes diphtheria, Corynebacterium diphtheriae, becomes pathogenic when it is infected with a lysogenic bacteriophage that carries the gene for the diphtheria toxin. Without the prophage, the bacterium won't produce that toxin and it's not harmful. So the virus replication and eventual lysis of the cell is what spreads it to other cells, and we end up with diphtheria. Another example is cholera, responsible for the symptom, Vibrio cholerae is the official term for the cholera toxin. It's encoded by a prophage within the bacterium, and lysogenic conversion allows this bacteria to produce the cholera toxin, leading up to the disease, very similar to the diphtheria one. So, grand summary. While both latency of animal and lysogeny in phages involve dormant phases, they differ in the host cells that they infect and then the end result for the cell that is infected. So, just to review, the lytic phases always end up in the cell death, and then the lysogenic phases ends up with the cell surviving even after infection. Lysogeny also plays an important role in the evolution of the bacterial pathogenicity through the transfer of virulence factors via the prophages. Thanks, guys.